Hello friends, welcome to CEC Live Lectures. Dear friends, today in this session we are going to continue in our series on cell biology, and we are going to discuss on plasma membrane. Though we have conducted one session on plasma membrane earlier, but as per the promises, we are here again, and to give you in-depth knowledge on the topic, we are going to discuss more. Friends, for the discussion on the topic, we have with us in our studios Dr. Charu Dogra Rawat. Dr. Charu Dogra Rawat is Assistant Professor in Department of Zoology, Ramjas College, University of Delhi. Dr. Charu Dogra Rawat is a prolific professor who believes in giving her best. Therefore, through the live platform of CC, she always exchange as well as give you information and knowledge on various topics. So, let's welcome our guest, Dr. Charu Dogra Rawat, and let's try to grab maximum knowledge through her. Hello, ma'am. Welcome to the lecture.
Thank you, Geetika. So, the most important part of a cell is the plasma membrane. The plasma membrane, which is basically, which makes a cell a cell. The sanctity of the cell is basically this plasma membrane, which keeps all the functions and every exchanges and all the activities which a cell performs is basically managed by this plasma membrane. So we'll talk about the plasma membrane today. We have already talked about the structure and the models of plasma membrane, a historical perspective that how we got to know the plasma membrane, which is now the structure of the plasma membrane, which we know as of now. And we also talked about the different components of plasma membrane, what makes a plasma membrane, the lipids, the proteins, and the carbohydrates. So today we'll start, because as we have to talk about the functions of the plasma membrane, but before we do that, we'll talk about the properties of the plasma or properties of these components. What role does they play and how they perform or help in the different kind of functions played by the plasma membrane for the sanctity of the cell. So the main objectives for today would be to understand the important property of plasma membrane, which is termed as fluidity. We talked about the different components, the lipids and the proteins, and we also talked about the recent most model, or basically the most accepted model, which is the fluid mosaic model, and which has this important component of fluidity of the plasma membrane. That is, that it's a dynamic structure. The lipids, the proteins, they keep on changing places, and it's not static. It's not that the plasma membrane components just remain where they are. So the fluidity of the plasma membrane is a very important property, and we will see and study in detail about this property. We'll talk about the fluidity of the lipids, and we will talk about the fluidity of the proteins. So talking about the fluidity of the plasma membrane and its components, gradually we'll move towards and understands deeply about the functions of the plasma membrane, the functions which it performs as the outer covering of the cell, as well as it performs as the covering of the different organelles which are present inside the cell membrane. Today we'll also take up an example of the erythrocyte, which is the red blood cell, and understand the plasma membrane structure through this example. So talking about the fluidity, we just have to understand the lipid bilayer. Each type of cell membrane has its own characteristic lipid composition, differing from one another in the type of lipids, the nature of the head groups, and the particular species of the fatty acyl chains. So in our previous lecture, we have already talked about the structural aspect of the plasma membrane, that how these lipid molecules are arranged. So we know that it is a lipid bilayer where the hydrophilic groups are basically outside, the polar head groups, and the hydrophobic chains are towards the inside. So it's kind of a bilayer which is present in the plasma membrane. Looking at the different components, we see that even the different parts of the cell membrane, or the different organelles carrying these cell membranes, contain different compositions of lipids molecules which are there. And this differs in the type of the lipid molecules, the nature of the head groups, as well as the particular species of fatty acyl chains. As you can see in this particular table, that there are lipid compositions of some biological membranes given. This is the phosphatidic acid and phosphatidylcholine. So you can see in erythrocyte there are around 90. This is percentage weight of the total lipid which is present in the cell. You can see in the human myelin, you can see in the beef heart mitochondria, and in fact in E. coli plasma membrane this is absent. Ethanolamine is the highest in beef heart mitochondria which is there, which is around 27, and E. coli has the largest weight percent, which is the 65% by weight total lipid which is present. So you can see that the different types of lipids, basically they are present in different amount in different plasma membranes, even within the same species or across the species. So it's not constant. It's not that every plasma membrane is made up of these components in these weight percent. It's not like that. So it varies amongst the different plasma membranes which is there. In fact, this is another table which shows the head group composition of the membranes of some mammalian liver cells, erythrocytes and nerve cells in weight percentage. So you can see here that again, in the erythrocyte of humans, the percentage of phosphatidylcholine is highest, then the phosphatidyl'inositol, serine, the phosphatidylethanolamine, inositol and sphingomyelin, and cholesterol is not present, and you can the cholesterol is around 20, the glycolipid, and you can see other components which are present here, and you can see the difference in the composition of these lipids in different organelles also. So in ER, in Golgi, in lysosome, the nuclear membrane, the composition is different, in neurons the composition is different. So lipid composition varies in different plasma membranes, in different species, and within a species, even in different cells, and within a cell in different organelles also. So it's not really static or established what is there. Another important thing is that there is an asymmetry across the membranes itself. So not as a total in plasma membrane, it varies between different organelles, but within an organelle, this bilayer lipid membrane, there is an asymmetry which is found. The lipid composition is not only different in different cell types and organelles, but also within the two leaflets of the same membrane. So we can see here that we know that it is a bilayer, the plasma membrane. So this is like, for example, the exoplasmic side, and this is the cytosolic side of the plasma membrane. And this example is again of a red blood cell or human erythrocytes, and you can see the differences in the composition. Towards the exoplasmic side, the sphingomyelin is very much in larger quantities. However, towards the cytosolic side, the ethanolamine or phosphatidylserine is in higher quantity which is there. So all the glycolipids of the plasma membrane are in the outer leaflet, where they often serve as receptors for extracellular ligands. So I would like to emphasize here that this structural arrangement of lipids across the two leaflets is very much correlated with the functions, and that is why when I started I said that we are going to talk about these properties, because ultimately it will form the basis of understanding the different functions of the plasma membrane. So when we talk about the different functions of the plasma membrane, we have to understand the different properties of the molecules or the components which makes up the plasma membrane which is there. So all the glycolipids of the plasma membrane are in the outer leaflet, where they often serve as receptors for the extracellular ligands. Phosphatidylethanolamine, as you can see, is concentrated in the inner leaflet, tends to promote the curvature of the membrane, which is important in membrane budding and fusion. So in here I have just mentioned some of the functions which we can correlate with the localization of these lipids across the leaflets, which we will talk again about the detail when we talk about the functions of the plasma membrane as such. So phosphatidylethanolamine is responsible for the curvature that occurs, which basically helps in cell fusion and cell budding, and it is too much concentrated towards the cytosolic side. Phosphatidylserine, again, as you can see, is concentrated in the inner leaflet. It has a net negative charge and physiological pH, which makes it a candidate for binding positively charged lysine and arginine residues, such as those adjacent to the membrane spanning a helix of glycophorin. So we'll talk about this glycophorin A protein in a while, but as you can see, this phosphatidylserine is higher because it's giving a kind of a charge, and we know that there is an electric potential across the plasma membrane which is maintained. This electric charge, this potential is again because of this asymmetrical distribution of certain lipids on the two leaflets. This phosphatidylinositol, it has a net negative charge at physiological pH, which makes it a candidate for binding the positively charged lysine and arginine residues. So, again a functional aspect which is very, therefore the property of this kind of arrangement is very important for the functions the plasma membrane performs. Again, you can see that the phosphatidylinositol which is concentrated in it's very lesser as compared to the ethanolamine and serine, but still it is more on the cytosolic side as compared to the exoplasmic side on the red blood cells. So can be phosphorylated at different sites on the inositol ring, which converts the lipid into a into a phosphoinositide. These phosphoinositides play a key role in the transfer of stimuli from the plasma membrane to the cytoplasm and the recruitment of proteins to the cytosolic phase of the plasma membrane. So this particular lipid molecule is a key molecule for the transfer of stimuli from the plasma membrane towards the cytoplasm. So this phosphoinositide, which it forms because of the conversion of the inositol rings, is actually responsible for taking the stimulus from the exoplasmic side and passing it on to the cytoplasmic side, where further signal transduction takes place and the stimuli give rise to a particular kind of a response. So again, this is because of the localization, asymmetrical localization of the lipid membranes across the two leaflets. Phosphatidylcholines and sphingomyelins, as you can see, are rather found on the outer leaflet, and as we said, they are mostly glycosylated and they act as the receptors for the stimuli or for the signals from the outside of the cell. The lipid composition is not even homogeneous within one monolayer leaflet. So we talked about that the different plasma membranes of different species, of different cells, and within cell different organelles, the composition is different. Then we said that within a cell or within a cell organelle even, or within a single plasma membrane, there is an asymmetry of lipid composition as far as the two leaflets are concerned. Now we say that even within one single layer or a monolayer of this bilayer, the lipid composition is not homogeneous. Lipids tends to segregate into domains due to phase separation phenomena. So we'll talk in detail about this phenomena, that how different kind of structures this lipid membrane can take place, and because of this aggregation, we have different kinds of structures that arise, which are again functionally correlated. So this is a very important aspect to understand before we move on to the functions of the things. Again, before we go, we need to talk about this one particular structure, which are called as the liposomes. An important feature of the lipid bilayer is its ability to self-assemble, which basically means that when a small amount of, for example, a phosphatidylcholine is dispersed in an aqueous solution, the phospholipid molecules assemble spontaneously to form the walls of fluid-filled spherical vesicle, which is called as liposomes. So, they can self-assemble into this bilayer formation, or a typical, in a very simple plasma membrane formation which is there. So when you put this particular phosphatidylcholine, for example, or any lipid molecule onto the water surface, or it is dispersed in an aqueous solution, it takes up the form of, or they assemble into such kind of structures which are termed as liposomes. So you can see it's a typical bilayer organization where the polar heads are towards the outside and the hydrophobic tails or the chains are towards the inside, and then it forms a very important hydrophilic core or the lumen which is there. So these structures are termed as liposomes, and the walls of these liposomes consist of a continuous lipid bilayer that is organized in the same manner as that of the lipid bilayer of a natural membrane. So basically, in a very stating in a very simple manner, we can actually simulate the plasma membrane as it occurs in the natural environment artificially by putting lipids onto the aqueous solution, and they will naturally arrange in that particular manner. So membrane proteins can be inserted into liposomes. A very important function of liposomes is that because now it forms the kind of natural plasma membrane environment, and when it goes, it can fuse with the plasma membranes, can adjust in there, and probably carry certain things in the hydrophilic region which it can transport. So these membrane proteins can be inserted into liposomes and their function studied in a much simpler environment than that of a natural membrane. That is one important application of liposomes. Another thing is that liposomes have also been developed as vehicles to deliver drugs or DNA molecules within the body. The drugs or DNA can be linked to the wall of the liposome or contained at high concentration within its lumen. So because particularly the hydrophilic molecules which we need to pass across the plasma membrane is very difficult, because the lipid plasma membrane is actually hydrophobic in nature. So a hydrophilic molecule had to pass across this lipid layer, and these liposomes can play an important role. So you can see here that certain the liposomes are tagged with antibodies, and these antibodies will target particular places where we need to go, and therefore some drug is crystallized in the aqueous fluid and they are stored here. And this is basically, so they can, the lipophilic drug can be stored in the bilayer, or the hydrophilic drugs can be put in the lumen and then added. Now, initial experiments with these liposomes failed because the immune system, took it as a foreign material and just destroyed it. So, the liposomes have been developed to carry this particular protective layer of, for example, polyethylene glycol or PEG. So, this is a model of a stealth liposome, as it is called as, and it has been used to deliver many anti-cancer drugs, etc. inside the cells for effective usage. So they are covered by this layer, and therefore it evades the human immune system or any immune system, and therefore you can deliver the target molecules into the desired cells or organelles if one wants to. So liposomes are very, very important in there. So coming back to when talking about again membrane lipids and membrane fluidity, the physical state of the lipid of a membrane is described by its fluidity or viscosity. So fluidity and viscosity, they are basically inversely related, because fluidity is basically the flow we are talking about, and viscosity when we talk about is the resistance. So higher the flow, lower is the resistance, lower the flow, higher is the resistance. So that kind of a thing. But we generally term as the membrane fluidity, that how fluid a particular membrane is. Now we have talked about the different compositions of lipid and that it is a very dynamic nature. Now we'll talk about what kind of movements that, or what kind of phase changes can occur in these lipid molecules that give rise to the membrane fluidity, and that ultimately give rise to all the different kind of functions a plasma membrane performs. Lipid membranes display melting transitions, which basically means that with a difference in temperature, there is a difference in the physical state of a plasma membrane. As you can see in here, this is the structure of the plasma membrane. The bilayer shown here is composed of two phospholipids, for example, the phosphatidylcholine and phosphatidylethanolamine. Now, above the transition temperature, so transition temperature is when one state converts into the another state, or one phase converts into the another phase. So at that particular transition temperature, you can see the lipid molecules and their hydrophobic tails are free to move in certain directions, even though they retain a considerable degree of order. So they are randomly moving. So there is still a kind of an order, but their chains are free to move. Below the transition temperature, the movement of the molecule is very much restricted, and the entire bilayer can be described as a crystalline gel. So in here, it's more of a fluid, but in here, it's more of a gel-like substance. So plasma membrane display these melting transitions. So at the transition temperatures, it can occur in these two physical states which is there, and that again explains this important property of fluidity of the plasma membranes, or the lipids of the plasma membrane as we are talking about. Lipid melting transitions depends on the chain length and the degree of saturation, that is the number of double bonds in the chain. The longer the chains, the higher is the melting point. However, double bonds in a chain drastically lower the melting temperature. So when we talk about this particular property, and because this property is arising due to the differences in the temperature, the factors which are responsible for higher transition temperature or lower transition temperature constitutes the length of the chain as well as the saturation of the chain which is there. You can see here the melting points of the common 18 carbon fatty acids chain. So you can see here it does not contain a double bond, it is 70, the melting point is here. So you can see that different fatty acids, as the saturation is increasing, the temperature is lowering. So basically lower the melting temperature, the double bonds in a chain drastically lower the, so 5, it has been drastically reduced from, for example, 70 to minus 54 because the length of the chain is constant here. So that reflects again a very important property of the physical state of the lipids, or how this fluidity can be modified. Physical state of the membrane is also affected by cholesterol. So apart from the phospholipids, cholesterol also plays an important role in the physical state of the plasma membranes. Cholesterol molecules disrupt the close packing of fatty acyl chains and interfere with their mobility, tends to abolish sharp transition temperature and creates a condition of intermembrane fluidity which is there. So in physiological terms, if we talk about, cholesterol tends to increase the durability while decreasing the permeability of a membrane. So we'll talk about more about the permeability when we talk about the functions of the plasma membrane, but structurally we can see that the presence of the cholesterol is basically disrupting the close packing of the fatty acyl chains and therefore creating a kind of interference with their mobility, and therefore they tend to abolish the sharpen change in the transition temperatures which is there. So flexibility of the membrane is reduced, and therefore high cholesterol is a kind of a threat, because when there is a high cholesterol deposition, the membrane becomes rigid. The membranes of the vessels become rigid. It's not flexible. They are not distensible. So when they are not distensible, the clots can occur. The flow of the blood is not as smooth as it is when the cholesterol level is low. So that kind of a thing, again, it depends upon how cholesterol influences the change, the transition temperatures of the plasma membrane which is there. Then phase transitions in membranes depends upon many more variables apart from the temperature, which is for example pressure, pH and the chemical potential of ions such as calcium. So pressure, for example, shifts the melting temperature towards higher temperature. The presence of a chemical such as ethanol, it lowers the melting temperature. So different factors or different variables are again imply their role on the fluidity of the plasma membrane. One important point to note here is basically that the lipid composition change in a coherent manner as a response to changes in the environmental conditions that can be expressed in terms of intensive thermodynamic variables. So what this word coherent basically means is that all these factors and variables are basically correlated in nature. They work in a very composite manner. So for example, the change in the temperature, whatever effect it gives, might also be given by the change in the pH, higher or lower, or the presence of certain ions, or the presence of certain chemicals which are there. So electrostatic differences, chemical differences, different kind of physical changes such as change in temperature and pressure. So they collaboratively, the environmental factors collaboratively or coherently defines the fluidity of the plasma membrane or the lipid composition, how it should be. And it is expressed in terms of different thermodynamic variables for understanding deeply into the aspect. Now, how does this membrane fluidity is maintained? The maintenance of membrane fluidity is an example of homeostasis at the cellular level. So this is again a very physiological response which is there, that it basically means that membrane fluidity varies among different membranes, and the maintenance of the membrane fluidity is because of this kind of homeostasis that needs to occur for the cellular level. Initial emergency response, for example, if a melting temperature is suddenly lowered down, or if a cell is suddenly put into a cold temperature, how does the membrane changes? So initial emergency response is mediated by the enzymes that remodel the membranes. Remodeling is accomplished, for example, in case of lowering of the temperature, as I said, and in response for making the cell cold resistant by, for example, desaturating single bonds in fatty acyl chains to form double bonds. This is accomplished by the enzymes called as desaturases. So desaturases will remodel the plasma membrane because now it is lying in a colder environment. They will create more double bonds. They will desaturate the single bonds and convert them into more double bonds. the reshuffling of the chains will take place, which is basically accomplished by phospholipases and the function of acyltransferases which is there between different phospholipid molecules to produce ones that contain two unsaturated fatty acids, which greatly lowers the melting temperature of the bilayer. So if a cell is placed into a colder environment, remodeling of the plasma membrane will take place. So that is what we talk about, the maintenance of the membrane fluidity. Because if a cell does not have these kind of adaptations or these kind of changes, then the cell will in a particular environment be very rigid and therefore will not perform its functions properly. So to attain that, the membrane adapts by changing its lipid composition or by remodeling the plasma membranes with the help of which is catalyzed by these particular enzymes and therefore become more adapted towards a particular environment. Additionally, the cell changes the type of phospholipids being synthesized in favor of ones containing more unsaturated fatty acids. So there is occurrence of more double bonds, there is shuffling of hydrocarbon chains that occur, fatty acyl chains that occur, and there is production of more of the fatty acids which are suited for that particular environment rather than the ones which were suited when it was not put in that kind of an environment. So the remodeling of the membrane is very, very important. Maintenance of fluid membranes by adjustment in such fatty acyl composition can be seen in the hibernating mammals, pond-dwelling fish whose body temperature changes markedly from day to night, cold-resistant plants and bacteria that live in hot springs, thermophiles. So that is how they suit to their environment by making changes in their plasma membrane. And this change can occur only because the plasma membranes are fluid in nature, dynamic in nature, and not a very static entity. Another important thing which we need to talk about is the lipid rafts. When membrane lipids are extracted from cells, although I must again state that there is a group which believes in the occurrence of lipid rafts, and there are groups which do not believe in the occurrence of lipid rafts. But we, it's my, I should explain this lipid rafts to you because functionally again, this structure plays an important role. So when membrane lipids are extracted from cells and used to prepare artificial lipid bilayers, cholesterol and sphingolipids tend to self-assemble into microdomains that are more gelated and highly ordered than the surrounding regions, consisting primarily of phosphoglycerides. These patches of cholesterol and sphingolipids are referred to as the lipid rafts. When added to these artificial bilayers, certain proteins tend to become concentrated in the lipid rafts, whereas others tend to remain outside their boundaries. So you can see here, these are basically the patches of sphingomyelin and the cholesterol which is there. And you can see these yellow spikes are basically the position of the GPI anchored proteins. So you can see these GPI anchored proteins are localized at these. So there is a disorder that has occurred because of the membrane transition, but still you can see certain order and accumulation of these lipids, particular lipids into the formation of such things which are called as the lipid rafts which is there. Lipid rafts are postulated to serve as floating platforms that concentrate particular proteins, thereby organizing the membrane into functional compartments. So again, this property related to the function, which basically is that the you can see, for example, here this is an example of a lipid raft. As you can see, it is a model of a lipid raft within a cell. The outer leaflet of the raft consists primarily of the cholesterol and sphingolipids. So you can see this is the outer part. It contains mostly of the sphingolipids, and the inner part contains of the this phosphatidylcholine molecules with long saturated fatty acids also tend to concentrate in the region. GPI anchored proteins are thought to become concentrated in the lipid raft. So as we have seen in this particular figure, you can see that GPI anchored proteins are concentrated on these lipid rafts rather than on the other structures. So this is basically functionally related and explains the functions of the lipids in a very effective manner which is there. So the possible movements these phospholipids can attain in this membrane, you can see here there is a transverse diffusion. So there is a flexing which is occurring, there is a lateral shift that occurs, and there is a transverse diffusion that can occur. So these are the different types of movements, and of course this flip-flop movement is the most restricted one. and in a subsequent session we will talk about the different barriers which restrict these movements and go into the movements of the proteins, how the fluidity of the plasma membrane is provided by the proteins which are there. Thank you.
With this note, thank you, ma'am. Thank you so very much for giving this productive session. Friends, you are requested to be with us as we are back after a short break. Thank you. Hello friends, welcome back to this session. Friends, as you know that today we are talking on plasma membrane, and for the discussion on the topic we have with us in our studios Dr. Charu Dogra Rawat. Dr. Charu Dogra Rawat is Assistant Professor in Department of Zoology, Ramjas College, University of Delhi. So friends, let's welcome our guest Dr. Charu Dogra Rawat once again, and let's try to understand the topic in detail. Hello ma'am, welcome to the session.
So we have been talking about the structure of the plasma membrane, the different components of the plasma membrane, and now we have reached to talking about the properties of the plasma membrane, and the most important property is the fluidity of the plasma membrane. We have talked about how the lipid composition is responsible for this fluidity, and now we move on talking about the membrane proteins, that how it inputs or provides the fluidity to the plasma membrane. Thereafter, we'll talk about a red blood cell, its membrane structure, trying to again, emphasize about the different or understand the organization of plasma membrane before we go into talking about the functions of the plasma membrane. So, talking about the membrane proteins, as we have seen, because lipids provide the matrix in which the integral proteins of a membrane are embedded. The physical state of the lipid is an important determinant of the mobility of the integral proteins. So we have already talked about this particular experiment, that how we know that the integral membrane proteins are fluid or they are moving. We have talked about this Fry and Eden's experiment where the cell fusion was taken up as an important experiment or as an important technique to reveal that the integral membrane or the membrane proteins are fluid in nature. The use of the cell fusion to reveal the mobility of the membrane proteins. So we talked about that there was a fusion between the human cell and mouse cell done by, for example, polyethylene glycol or Sendai virus or anything, and these two are fused together. The different proteins are tagged by different fluorescent antibodies, and when they are mixed together, initially you can see in this particular micrograph also that there is a difference, there is a separation of the red fluorescent proteins and there is a separation of the green fluorescent proteins which is there. So initially there is a separation of these tagged proteins, but after around 40 minutes they became homogeneously or they became distributed all along the entire fused cell. So definitely this particular technique pointed out to the fact that the membrane proteins are also fluid in nature because the matrix is fluid, the membrane proteins are also fluid, and they basically go from they kind of flow from one place to another, or undergo or show certain movements. Again, this is a very fluid statement to state, because this fluidity is again restricted by different barriers. So in the last session we only talked about the lipids fluidity, we haven't talked about the barriers, because once we understand about the fluidity of both the components, we'll talk about the certain barriers which restrict the movement of both the lipids as well as the proteins which are there. So cell fusion is an important technique to reveal the mobility of membrane proteins. Other techniques such as fluorescent recovery after photo bleaching, called as FRAP, or single particle tracking, which is SPT, is also performed, and it suggested that some membrane proteins move randomly throughout the membrane protein. So this is now very important aspect, understanding the mobility of the proteins through these two different techniques. And you can see here the different kinds of protein movements, the patterns of movements of integral membrane proteins which are there. So certain proteins, for example A here, they move randomly throughout the membrane, though generally at rates which are considerably less than would be measured in an artificial lipid bilayer. So the diffusion is slower, although the mechanism or why it is slow is not really elucidated, but the diffusion, a particular diffusion, is again slower, but they move randomly throughout the plasma membrane. Then there are certain proteins, some membrane proteins fail to move and are considered to be immobilized. So proteins such as B here, they are immobilized protein, they can't move. So certain proteins can move randomly and certain proteins just can't move. In some cases, a protein is found to move in a highly directed, that is non-random manner toward one part of the cell or the another. For example, you can see here the protein C. It is moving in a particular direction, which is maybe say away from the outer membrane or whatever, but it is moving in a particular direction. It's a non-random kind of a movement which is there. So random movement, immobilized proteins, and directed proteins, or the non-random movement of the proteins is there. The largest fraction of the protein species exhibit random or Brownian movement within the membrane at rates consistent with free diffusion, however, over a very short range, which is a few tenths of the micrometer. So the major kind of movement shown by the integral membrane proteins is the Brownian or the random movement which is there. But this is usually at a very, very short range, which is 1/10 of a micrometer that is there. Long range diffusion occurs, but at slower rates, apparently because of the presence of the system of barriers. So the random movement of the integral proteins is too much. Then lipid movement we have already talked about, but particularly these proteins, there are certain barriers which does not allow them freely to move around. And what are these barriers what we are talking about? The major barriers, one kind of thing is that some proteins are really crowded. They are so close to each other that restricts their movement. So some membranes are crowded with proteins so that the random movements of one molecule can be impeded by its neighbor. For example, you can see here the protein D. These proteins are too close to each other, and there is not much space to be randomly moving around. So that is one kind of a barrier that occurs in the proteins, particularly those membranes where the concentration of integral membrane proteins is very high. Then the most important barrier for the protein movement is the underlying skeleton that forms a network of fences around portions of the membrane, creating compartments that restrict the distance an integral protein can travel. When we talk about the shorter distances or shorter diffusion, then one important barrier that plays an important part is this underlying matrix which we see. So this underlying matrix is actually restricting this integral membrane proteins path which is there. It can move around where you have these kind of fences, and there are gaps in the fences. Only through that these integral membrane proteins can move. Otherwise they have a very restricted environment where they can move, and this kind of, you can see these blue regions, they are creating the sort of fences which is there, and these are underlying proteins, the skeleton network which is formed. The movement of a transmembrane protein through the bilayer is slowed by extracellular materials that can entangle the external portion of the protein molecule. So another barrier is that, for example, there are transmembrane proteins. So towards the cytosolic side, there could be certain things which are attached to these particular proteins. And because of these things which are attached to this particular protein, its movement will be restricted. So you can see the different patterns of movements of integral membrane proteins that is occurring in here. Apart from this, when we talk about the peripheral proteins, or the concentration or aggregation of proteins, as we were talking about the concentration and aggregation of lipids, then there also again occurs the specific domains, and there is cell polarity that can be found. So most membranes exhibit distinct variations in protein composition and mobility, especially in cells whose various surface display distinct functions. So again, this property of fluidity and mobility, as well as the presence or absence of certain proteins, is related to the functions that plasma membrane performs. In a similar manner as when we talked about the lipid composition and the lipid mobility that we talked about. So this particular, for example, in here there is a there is a plasma membrane of an epithelial cell, and you can see that this epithelial cell has three different plasma membrane regions. One is the apical plasma membrane that regulates the nutrient and water intake and regulates the secretion, and as well as it is important for the protection of the cell. This is the lateral plasma membrane, which is important for the cell-cell contact and adhesion and cell communication, and this is a basal membrane which is basically performs the function of cell substratum contact or generation of ion gradients. The apical surface of this intestinal epithelial cell contains integral proteins that function in ion transport and hydrolysis of disaccharides, such as, for example, in sucrose and lactose. The lateral surface contains integral proteins that functions in intercellular interactions, and the basal surface contains integral proteins that function in the association of the cell with the underlying basement membrane. So you can see that why we started talking about the property, this important property of different composition as well as different fluidity of the plasma membrane components, is because they are directly related. It's a vice versa thing related with the functions of the plasma membrane. So in this particular example, a very good example where you can see three different types of plasma membrane, and these three different types of plasma membrane in a single cell, the composition of the proteins is different because the functions they are performing is different, or the composition is different and therefore they can perform different functions. It's correlated thing. So the property or the structural components of the plasma membrane as well as the fluidity of the plasma membrane largely depends upon the functions a particular plasma membrane is performing. In here, the most diverse or differentiated plasma membrane is that of a mammalian sperm. So you can see in here, as revealed by the fluorescent antibodies, these are the three different pairs of micrographs which shows the presence of different proteins at different parts of the sperm. So there is an anterior head, there is a posterior head, and there is a posterior tail. When antibodies are used specifically against certain proteins, you can see that one kind of protein accumulates towards the head. Another kind of proteins accumulates at the posterior head region. Another type of protein is basically accumulating at the tail region. So same plasma membrane of the same cell is showing the concentration of different proteins at different parts of the same plasma membrane, and this is because they perform different kind of functions. Tail performs a different function. Head consists of something else, and it needs to penetrate the zona pellucida, and therefore it needs to fuse with the plasma membrane of the egg. Therefore, the proteins which are present on the head will be different on the proteins which will be present on the tail, whose primary function is to provide energy by propelling itself so that the sperm can move, and the different proteins which are present at the posterior head where the centrioles will be present. So this is the highly differentiated differentiation of membrane proteins that occur in the plasma membrane is revealed. So I hope you understand this point by this time, that the composition of the membrane as well as the fluidity of the membrane varies and it is correlated with the function that plasma membrane performs. Before going on to the functions of the plasma membrane, we will take up the red blood cell as an example of plasma membrane structure. And I think after I finish with this plasma membrane structure, it will be really engraved in your minds that how and why this plasma membrane structure is very important. So we talked about the different models, we talked about the different components, and we talked about the different properties of these components, and taking this example, it will kind of sum up this entire knowledge what we have gained, so that in our next session we can apply this knowledge towards the understanding deeply the function of the plasma membrane, which again is very important. Plasma membrane is the most important part of a cell, as I always say. Therefore, understanding the plasma membrane is foremost and most important thing for understanding the structure and function of a cell as a whole. So let's take red blood cell as an example of plasma membrane structure. Reasons for the popularity of this membrane, why we study this plasma membrane, is that because the cells are inexpensive to obtain and readily available in huge numbers from the whole blood. You can just isolate them from the whole blood samples. They are already present as single cells, because if we want to take the cells from any other tissue, we need to dissociate the tissue, or any other organelle, we need to dissociate their organelle, we need to dissociate the tissue into single cells, and then we can work on these single cells. But red blood cells, they are already single cells and they need not to be dissociated from a complex tissue. Another reason why we use red blood cells is that they are lacking most of the organelles. They lack nuclear and cytoplasmic membranes that inevitably contaminate the plasma membrane preparation from other cells. Because we are now studying about the outer plasma membrane structure, because they lack different organelles, therefore the cross contamination of other membranes is not there. And we can exclusively talk about the outer plasma membrane of a particular cell, and therefore it's much more simpler to study and devoid of any contamination of plasma membrane from other organelles. Then the purified intact erythrocyte plasma membranes can be obtained very easily and very simply by placing the cells in a hypotonic salt solution. So when you place the cells in a hypotonic salt solution, the water just goes inside, the cell swells, and there occurs kind of leakage. So this leakage will remove most of the hemoglobin, and therefore what you are left with is the outer plasma membrane, which is transparent in nature. So it gets devoid of all the pigment very easily. Just you place the red blood cells in a hypotonic solution and you will get an intact purified plasma membrane in no time. So you can see these scanning electron micrographs of human erythrocytes. This is the micrograph showing plasma membrane ghosts. This is the so when you put these cells in a hypotonic solution, they will lose the red pigment, and all you can see is this kind of a structure, which is basically the can then be isolated. After this, these are hemolyzed and swelled. So very easy to purify the plasma membrane molecules. Now, once erythrocyte plasma membranes are isolated, the proteins can be solubilized and separated from one another or fractionated using what is called as the STS page, polyacrylamide gel electrophoresis, in the presence of the ionic detergent sodium dodecyl sulfate. Now, this sodium dodecyl sulfate gave a negative charge, a homogeneous negative charge to the plasma membranes, and then when they are run on this polyacrylamide gel, the different components separates according to their molecular weight, because the net charge on all the components is basically negative, so they kind of separate. So you can see basically this SDS page gel, the major proteins of the erythrocyte membrane are separated into about a dozen conspicuous bands. So you've got the membrane, you just take the membrane, you solubilize the proteins and separate it. So you run a SDS page gel. So when you run this SDS page gel, you see that these are the peripheral proteins and these are the integral proteins. We have identified it with the other manners, but otherwise you can see all these different kind of bands which you can be there. This is spectral. We'll talk in detail about all these proteins now, which are the component of the structure of the plasma membrane which is there. So this is the model of the erythrocyte plasma membrane as viewed from the internal surface. So you can see this is the red blood cell and this is the structure which we have attained. This is a bilayer structure. If you look at this particular molecule, this is a single phospholipid molecule. These are the hydrophilic chains, these are the hydrophobic chains. So this is the lipid bilayer, the light blue color which is there, and these are the different membrane proteins which are there. So some of the proteins are integral membrane proteins. As you can see, they are embedded, and some of them are even spanning. They are transmembrane proteins which are there. So these are the integral membrane proteins or transmembrane proteins which you can see here. They can be found here, and these proteins which are on the top are basically the peripheral proteins which is there. So this is the lipid bilayer, integral proteins, and the peripheral proteins that you can see. So this is the model of the erythrocyte plasma membrane, and we'll take up one by one these proteins and look at their functions that makes the the erythrocyte plasma membrane what it is. The most abundant integral protein of this membrane are a pair of carbohydrate containing membrane spanning proteins called as the band 3 and the glycophorin A. So you can see that this particular protein, blue color, is the band 3 protein, and this green color protein is a glycophorin A protein, and this is the most abundant integral membrane protein which is found in the plasma membrane of the erythrocyte. Band 3 is present as a dimer composed of two identical subunits, a homodimer. Each subunit spans the membrane at least a dozen times and contain a relatively small amount of carbohydrate, which is 6 to 8% of the molecule's weight. So band 3 is a homodimer consisting of at least a dozen times spanning the entire plasma membrane which is there. Band 3 protein serves as a channel for the passive exchange of anions across the membrane. Now we know that the primary function of the blood is basically to carry oxygen from the lungs to the different parts of the body and carry back the the bicarbonate ions to the lungs. So this anion exchange occurs at the tissue level, and this anion exchange occurs on the plasma membrane, and the entry and exit of these different kind of anions occurs through these channels which are formed by the band 3 proteins on the plasma membrane which is there, because it has to take up or load the bicarbonate ions and unload the oxygen ions, and therefore the same way it has to take up the other things or at the level of lungs or at the level of other tissue parts. So this kind of a passive exchange of anions takes place through the channels of the band 3 which is there. Now glycophorin A, which is again abundant integral membrane protein found in the plasma membrane, is again present as a dimer. Each subunit, however, spans the membrane only once, and it contains a bushy carbohydrate cover consisting of 16 oligosaccharide chains that together make up about 60% of the molecule's weight. So this green one, it just spans the plasma membrane once, and you can see that all these yellow are the oligosaccharide chains which are there. So glycophorin is a glycoprotein, and this glycoprotein is the name is also glycophorin A, although there are other glycophorins which are also present, the glycophorin B, C, D, but glycophorin A is the most abundant one, and you can see that there are carbohydrate chains which are associated with this integral protein on the red blood cells which is there. Primary function of glycophorin derives from the large number of negative charges borne on sialic acid, the sugar residue at the end of each carbohydrate chain. Because of these charges, red blood cells repel each other, which prevents the cells from clumping as they circulate through the body's tiny vessels. So you can see that this membrane, this is the inner membrane part and this is the outer membrane side. So on the outer membrane, this glycophorin part consists of all these oligosaccharide residues, and the terminal residue of the oligosaccharide is a sialic acid. So this sialic acid gives it a negative charge. So the outer membrane of the red blood cells are negatively charged. So one red blood cell will repel the other red blood cell because of the occurrence of these negative charges, and therefore avoids what is called as a clumping of the red blood cells. So it has been seen that if this glycophorin A is not present, then you cannot see that clumping occurring, because in that case the band 3 proteins are highly glycosylated. So the glycosylation definitely is causing the red blood cells to occur ideally, although when glycophorin A is not there, then the band 3 becomes highly glycosylated and performs a similar function as the glycophorin A which is there. But this negative charge is important for keeping the cells intact and individual rather than clustering or clumping together. Glycophorin also happens to be the receptor utilized by the protozoan that causes malaria, providing a path for entry into the blood cell. So as we have already seen, these integral membrane proteins contains these oligosaccharide residues which are receptor for a lot many things. They are also receptor for the malarial parasite. And the one, so the individuals which lack glycophorin A are basically saved from the malaria also. So these are the two very important integral membrane proteins which are found. Coming to look at the peripheral proteins which are there. The peripheral proteins of the erythrocyte plasma membrane are located on the internal surface and constitute a fibrillar membrane skeleton that plays a major role in determining the biconcave shape of the erythrocyte. So you can see that the inside is basically this biconcave in nature, and this biconcave nature is basically by the collaborative action of these peripheral proteins which are present towards the inside. And this is also giving a kind of a network. The sanctity of the cell is because of this kind of a skeleton which is provided by this peripheral proteins that are there. So we'll talk about these proteins, encheirin, spectrin, actin, tropomyosin, that how they give the shape to the red blood cells and how they basically stabilize the plasma membrane by giving a skeletal structure for the other integral membrane proteins that can be found. The major component of the skeleton is an elongated fibrous protein called as spectrin. Spectrin is a heterodimer approximately 100 nanometers long, consisting of two units alpha and beta that curl around one another. Two such dimeric molecules are linked at their head ends to form a 200 nanometer long filament that is both flexible and elastic. So you can see this is the spectrin molecule, and these two subunits are actually twisted on each other. So this one molecule and this is the second molecule, and therefore they kind of form a kind of a network which you can see here. So these are the spectrin molecules which are there. Spectrin is attached to the internal surface of the membrane by means of non-covalent bonds to another peripheral protein which is called as encheirin, which in turn is linked non-covalently to the cytoplasmic domain of a band 3 molecule. So you can see this is the spectrin molecule. These green globular structures are the encheirins. So these spectrin associates with the encheirin, which finally associates with the C-terminal domain of the band 3 protein. So this is the band 3 protein. This is the band 3 protein which is there. So they stabilize by interacting with the cytoplasmic side of this particular integral membrane protein which is there. So the spectrin, encheirin and band 3 protein which is there. Spectrin filaments are organized into hexagonal or pentagonal arrays. This two-dimensional network is constructed by linking both ends of each spectrin filament to a cluster of proteins that include a short filament of actin and tropomyosin proteins typically involved in contractile activities. These electron micrographs show the arrangement of the proteins of the inner membrane skeleton. So you can see here that this kind of a hexagonal or pentagonal arrays that occurs is because of the interaction of the spectrin filaments. They arrange or organize in this kind of a manner, and in this way they also include the short filaments of actin and tropomyosin. So actin and tropomyosin, we know is basically they are present in muscles also. So this actin and tropomyosin is basically for the flexibility or elasticity of the plasma membrane which is there, and it provides the energy for the contractile functions of the plasma membrane. So actin and tropomyosin also interacts, and therefore they kind of arrange in such a hexagonal or pentagonal manners which is there. This spectrin actin network gives the cell the strength, the elasticity and the pliability necessary to carry out its demanding function. So the spectrin actin network gives the cell the strength, the elasticity and pliability necessary to carry out its demanding function which is basically there. So this basically tells, we have talked about the the structure of the plasma membrane. We have talked about the different models of the plasma membrane. We talked about the different components of the plasma membrane, which are primarily the lipids and the proteins. And in today's session, we very importantly talk about the property of of these components, even the composition, the number or the presence of these particular lipid molecules or protein molecules in different plasma membranes, as well as we also talked about the fluidity, how these components give rise to the fluidity of the plasma membrane, which is also the mosaic nature as well as the fluid nature we talk about in detail. And we looked at the example of a red blood cell as an erythrocyte as the membrane structure. Now having a good knowledge of the properties of the plasma membrane, we can now very easily understand the functions that this plasma membrane performs, that we are going to take up in a next session. Thank you.
Thank you, ma'am. Thank you so very much for your inputs to the lecture. And friends, we believe that your feedbacks are important for us. So do write to us at info.cec@nic.in. The lecture is going to be uploaded on YouTube soon. Keep watching us, keep writing us. We will be meeting. Till then, take care. Goodbye. Thank you, ma'am. Thank you so much.